In this particular series, I'm going to mention to you and highlight the differences between prokaryote and eukaryote. But before we dive into the differences, I would first of all like you to know the difference between prokaryote and eukaryote. And looking at prokaryote, these are simply organisms that do not have a true nucleus, and the DNA lies freely in the cytoplasm and they are not being enclosed by membrane. On the other hand, eukaryote simply means that these are organisms that simply have a true nucleus, and the DNA, of course, are being surrounded by membranes. But prokaryote, the DNA lies freely in the cytoplasm without being surrounded by membranes. Now, I'll state to you the various and major highlight differences between these two, prokaryote and eukaryote. First, I'll share with you a formula which I use to see how we can easily come out with the differences and memorize this easily. And I'll share with you this formula. And the formula we are going to be using to easily outline the major differences between these two is called FOPC-gen. That is FOPC, which is FOPC, 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 then gen, which is GERN, FOPC-gen. I'm going to use this particular formula to easily highlight some of the major differences between prokaryote and eukaryote. Also, I would like you to know that the first three letters of this formula FOPC-gen are all square. That is FOP are square, F square, O square, and P square. And the third letter, which is C, is cube. That is C cube. And the rest are single. In other words, FOPC-gen, that is FOPC-GERN, the F is square, the O is square, the P is square, and the C is cube. Now, let's look at the differences. I'm going to begin with the first, which is square. The square simply means two features, which is going to start with F. That is two features which are going to start with F. So let's begin with the first one. The first F here is going to stand for form. Form, that is FORM, the form. And looking at prokaryote, they are mostly unicellular. In the term of the form, prokaryote are unicellular. In other words, they have mostly one single cell. And looking at that of eukaryote, they are multicellular. In other words, multiple cells. So the first F means form, which is for prokaryote, they are mostly unicellular, and for eukaryote, they are multicellular. The second F here will stand for flagella, and for prokaryote, they have a simple flagella, and they do not have any microtubules. In other words, they are lacking microtubules. Looking at eukaryote, they have a complex flagella with a 9+2 arrangement, and they have microtubules. In other words, for prokaryote, they don't have microtubules, and eukaryote have microtubules. So the next F here, which is the flagella, this is the difference. For prokaryote, they do have a simple flagella, and of course, without any microtubules. On the other hand, the flagella is complex, and with the 9+2 arrangement, they do have microtubules. Now, moving to the next letter, which is O, we have O square. In other words, O for two of these features. The first O is going to stand for organisms. And looking at this feature, we want to look at organism, the difference, prokaryote and eukaryote. Looking at the first O, which is organism, we want to look at which organism fall under prokaryote. Of course, the organisms that fall under prokaryote are mostly bacteria. But looking at that of eukaryote, organisms that fall under these are mostly fungi. We have animals and protista. There are four main organisms that fall under eukaryote, and for prokaryote, just one. For prokaryote, we have bacteria, but for eukaryote, we have four of them. One, we have protista. Next, we have plant. Next, we have fungi. And the next, we have animals. These are the organisms. So the different organisms here have been differentiated for prokaryote and eukaryote. The next O here will stand for organelles. Let's look at the difference in the organelles between these two. Of course, looking at prokaryote, they have few organelles, and none of these organelles is being surrounded by membrane or envelope. Looking at the next, which is eukaryote, they do have many organelles, and they are being surrounded by membranes or envelope. So this is the next feature which we use to make the difference between prokaryote and eukaryotes. So now we are going to move to the next, which is P, and we also have P, which was square. You remember the term we use as a formula, oxygen. So now we are on P, and this P is square. Therefore, we are going to use two features of P. So the first P here is photosynthesis. So looking at photosynthesis for the first P, we are going to say that prokaryotes, of course, do not have any chloroplast, and therefore photosynthesis instead take place on membrane that do not have any stackings. But on the other hand, for eukaryotes, they do have a chloroplast, and photosynthesis take place on membranes which have been stacked into lamella or grana. Therefore, to look at this difference here based on photosynthesis aspect, of course, photosynthesis take place on membrane for prokaryotes that do not have any stacking. But for eukaryotes, the membranes where photosynthesis take place have stackings, and it has been stacked into lamella or grana, and they do have a chloroplast. But that of prokaryote have no chloroplast. So no chloroplast for prokaryote, but for eukaryote, they do have a chloroplast. The next P will be protein synthesis. Why both of these two, that is prokaryote and eukaryote, synthesizes protein, the difference here is the fact that for prokaryote, they actually synthesize 70S ribosomes, which is smaller. But for eukaryote, it is 80S ribosomes, which is larger. Therefore, the ribosomes being synthesized by prokaryote, which is the protein, is 70S based on size. That is smaller size, 70S, but that of eukaryote is 80S, which is larger. So in terms of the feature of protein synthesis, the proteins synthesized by prokaryote are smaller, that is 70S, but that for eukaryote are larger, which is 80S. Now, looking at C of the formula, which we stated that oxygen, we have C cubed. In other words, we have three main features under C. So we are going to begin with the first C. The first C here is going to stand for cell division. The first C here is going to stand for cell division. And of course, looking at the difference, for prokaryote, mostly they undergo binary fission, and there is no formation of spindles. But on the other hand, for eukaryote, cell division here is actually meiosis and mitosis, and spindles are being formed. The next C here will be cell wall. Of course, looking at the cell wall and making the difference between prokaryote and eukaryote, I want to begin at prokaryote. The cell wall of prokaryote is rigid and contains polysaccharide with amino acid. But here, the cell wall is being strengthened by murein. That is murein. On the other hand, for eukaryote, the cell wall here is also rigid, containing polysaccharide. But in the case of eukaryote, especially plant cell, the cell wall is being strengthened mostly by cellulose. And for fungi, the strengthening material for the cell wall is chitin. But for animals, of course, they do not have a cell wall. So now looking at the difference in terms of the C here, we stand for cell wall. For prokaryote, you're going to see that the cell wall is rigid, containing polysaccharide and amino acid. And here, what actually gives the strength of the cell wall for prokaryote is mostly what we call murein. But that for eukaryote, for plant cell, the main strengthening material is cellulose. And of course, the cell wall is also rigid, having polysaccharide. But the main strengthening material for plant cell wall is cellulose, but that for fungi is chitin. But animals, on the other hand, have no cell wall. The next C here will stand for the cell size. For prokaryote, the measurement is between 0.5 to 10 micrometer in diameter. But that of eukaryote, it is 10 to 100 micrometer in diameter, which is about 1,000 to 10,000 times more than that of prokaryote. So of now, we have 9 different features we have listed following this formula oxygen. And the first 4 letters, which we have square or cube, that is the first 3 as square, making 6 features, and the third, which is C, as cube, adding another 3 features with a total of 9 features we have listed. In the next series, we are going to mention the remaining features for GERN, GERN, and what are these features and the differences for prokaryote and eukaryotes. To those of you who want to receive updates on new uploads based on the new tutorials we are going to be uploading, then of course you want to make sure that you subscribe and enable the notification bell icon as well as liking this video so that you are going to receive new notifications and updates when the new video will be uploaded. Also, get ready and be prepared for the quizzes we are going to set in the upcoming video so that we are going to test you on the knowledge of these so that you stay up to date on what we have shared. Do not forget to like, comment, and subscribe, and see you again in the next series. And bye-bye for now.